দাও না।
conjugation type.
হ্যাঁ।
This conjugation type. So in case of meiocore, it appears like this. Two meiocore mycelium are found parallel to each other. Gradually, enlarge in size, and for the first time in the life cycle of meiocore, septum. Listen carefully, is most important for the answer. Septum is developed. So in phycomycetes, septum development occurs during sexual reproduction, that is during conjugation. So it's the most important question for any MCQ test. You see, this is the septum. Septum development is also a septum. Now, the anterior part, the part of the septum, you will get some gametes are produced. This part is called together gametangia.
তাই করো।
And the cell just be the septum, and this side is called, remember this thing, suspensor. The common question coming in that, the suspensor. And the gametes produced here, either it is or it is, these gametes are called cynogametes. Cynogametes means multi-nucleate gametes. Cynogamete development is possible. Why cynogametes are produced here? Again, another most important, you make a start here. Why cynogametes are produced here in phycomycetes? Because their mycelium is cynocytic mycelium. As the mycelium is accepted cynocytic in nature, hence cynogametes are formed. This question is coming in the answer. Make a star and write this. As the mycelium is accepted cynocytic, hence cynogametes are formed here. Then comes to the next. Now they are linked. And in the next diagram, we will see that common gas dissolves, and here we will get all gametes together. After that, what happens here you see, this plasmogamy occurs. You can call it plasmogamy. What is plasmogamy? Union of cytoplasm of gametes. This is called union of cytoplasm of gametes, and this is called a plasmogamy. After that, now what happens you see, so here you will get, even I am showing at least two gametes like this. So here is called karyogamy.
Eventually, the synozygous spores means multinuclear zygospore. Sometimes question comes, the diploid cell produced as a result of sexual reproduction in meiochore. Listen carefully. Diploid cell produced in sexual reproduction in meiochore is called, as some options, zygote, oospore, and zoospore. Out of these four options, which one should be correct? Raise your hand. In the sexual reproduction of meiochore, the diploid cell produced is called zygote, zygospore, oospore, and zoospore. Which one is correct? Anyone please raise your hand. No one. How can it be possible? Orko Prabha, do you hear me?
Yes, sir.
Bolo, which one is the answer?
Sir, Janinath, sir.
What I said earlier, I think in my earlier classes, that whenever diploid cell produced by means of conjugation is called zygospore. Diploid cell produced by fertilization is called zygote. Write it down now. Write it down, please, everyone. Diploid cell produced by means of conjugation. Diploid cell produced by means of conjugation is called zygospore. Next slide. Diploid cell produced by means of fertilization is called zygote. Clear?
Sir, please repeat.
The diploid cell produced by means of conjugation is called zygospore. And the diploid cell produced by means of fertilization is called zygote. Because here what type of reproduction occurs? What type of sexual reproduction? It is conjugation. It is conjugation. So product of conjugation should always be zygospore, you should remember it. And product of fertilization should always be zygote. Now after that pseudozygospores are formed, then pseudozygospore undergo meiosis to produce haploid spores, and they will give rise to neomycelium. This is what about the sexual reproduction in phycomycetial fungi. That means it appears like this. Look, this is called germ sporangium. So, gynozygospore on germination give rise to germ sporangium. And inside the germ sporangium, meiosis occurs. Meiosis occurs here to produce the germ spore. So, they are called germ spores. And eventually, due to the breakdown of the wall of the germ sporangium, germ spores are released, germ spores, and they give rise to neomycelium. It is happening here. So, germ spores are producing meiosis, meiosis occurs in the germ sporangium. You also need to remember this thing. The germ sporangium. So this is what about the life cycle pattern found in phycomycetial fungi. Now we proceed to some economically important such fungi. Dalia to mute karo. Dalia mute your audio. Now we are proceeding towards the Phytophthora infestans. Causes late blight of potato. What is late blight of potato? You see, if it is a potato leaf, you will get some black patches towards the periphery. This is the blight. The cells and tissues are dead there. In case of tumors, if it is a potato tuber, here also you will get some patches. That means these cells are actually destroyed, that become dead. So this is going to call it infected leaf, the infected tuber. Next one, Albiogo candida causes white rust in crucifers. Crucifers, crucifers refers to the plants belonging to the family Cruciferae, which is now called Brassicaceae. Common crucifer is mustard. Everyone knows mustard plant. So what rust in crucifers? Actually, what happens here, if it is a leaf of crucifer like this, then you will see some white patches. So there is white patches all over the leaf surface. Then this is called white rust in crucifer. And the organism is Albiogo candida. Peronospora parasitica causes downy mildew in, suppose, pea, suppose sometimes like mustard, etc. What happens in downy mildew? In case of downy mildew, woolly appearance found on leaves. The leaves show some woolly appearance, then this is called downy mildew. What a minute, please. This is the downy mildew.
Yes, sir.
Now, some other phycomycetial fungi I am to talk. Which one is called?
Sir, it is a phone number of phycomycetial fungi.
1, 2, 3, 4. Black bread mold, Rhizopas. Why it is called so? If after purchasing bread from the market, if you leave it for some 2-3 days, then you will see such molds. I am showing you 1 minute. Right?
Yes, sir.
You know, on
Yes, sir.
it's commonly seen in the bread. Now you see, here density increases. You can check it. Don't touch it, but you can check it by leaving some bread piece outside the house. Not actually, and so that actually it is not rain fed actually. So this is what about the black bread mold. Initially green, now you see huge structure developed of the black bread mold. Next comes to the another one. Dung mold. It grows on dung. This phycomycetial fungus grows on dung and is called dung mold. Example, Mucor. Mucor is also called dung mold. Mucor is again called Coprophilus mold. Do you know the meaning of the word coprophilus? Do you know what is coprophagy? You see, some animals like guinea pig, what is found in them, the fecal matter they are to release at night. It is soft and mucus mixed. And guinea pig used to take it again. We have to say eat it again. And this nature is called coprophagous nature. Feeding on dung, feeding on fecal matter. So any organism which is feeding on fecal matter, the term used is coprophagy, as it loves to grow on fecal matter. As mucor loves to grow on fecal matter like dung, it is also called coprophilus fungus. Most important term, you need to remember this. Coprophilus mold. Which one? Mucor. Why? Because sometimes question in 1 year in PMT, I think question comes like this. Mucor is called Coprophilus mold because point 1, it grows on bread. Point 2, It loves to grow on dung. Point 3. Eating and the mucor is eating its own fecal matters. Point 4. None of this. So if it is not clearly known, then it will be difficult to answer. That's why while I am putting stars in any part, you need to remember it very carefully. Because such type of questions are coming in the NEET. And that's why I am putting stars whenever I am giving such questions. So this is about the dung molds. Then Pilobolus is another. Pilobolus is also called dung mold. What is the importance of this? Why I am writing Pilobolus here? Sometimes question comes, in 1 year it comes like this. The dung mold is or are given mucor, given Pilobolus, given Rhizobus, given Phytophthora. So Phytophthora, no question of it. Now there is a question. Whether mucor or Pilobolus, and then another option should be there, both these options. So in that case, remember this thing, Pilobolus and mucor, both are dung molds. So these are the important phycomycetial fungi. Now we are proceeding towards the Ascomycetes. Ascomycetes. Next group. So here, most important first point, mycelium is septate. Mycelium is septate mycelium. Earlier in the Phycomycetes, we have seen mycelium is aseptate. I think you can remember. But here the mycelium is found to be septate. The cell wall is made of chitin. Orpita, tell me.
Chitin is nitrogen containing polysaccharide.
Okay. Basically, it's a polysaccharide having nitrogen. So remember this thing. So it is also other name of which is fungal cellulose. Most important thing, most important point, you need to stir it. Motor structures are not found. In the life cycle of Ascomycetial fungi. The most important thing, we get some motor structure in case of Phytophthora infestans, which are called juice pore. I discussed it. So mycelium is septate, cell wall is made of chitin, motor structure asexual. So here the main asexual mood conidia. So here asexual reproduction takes place by means of conidia. What about this conidia? You see, So, Conidia. It's called conidia. Now you know what are conidia. Write down, please. Conidia are asexual spores. Conidia are asexual spores produced exogenously in chain. Exogenously in chain. These structures are called medulla and this is sterigma. Sterigma. This is the mycelium. So in the conidia formation, in penicillium, I am showing you a single branch, but you know in penicillium, it is actually a lot of branches are developed. Remember this thing. It appears in mycelium, or you can call it, better to say, conidiophore. Structure bearing conidia called conidiophore. You can write structure bearing conidia called conidiophore. But it is found to be highly branched actually. If I am showing you again, a lot of medullae found. Again, from here, some branches may develop. Like this. And you will get the conidiospores like this here. This is what happens in case of penicillium actually. From each medulla, a number of conidia may be developed. So, this is what about the asexual reproduction in ascomycetial fungi. So, one conidiophore may give rise to two or more conidia, more medulla. From each medulla, again it can develop two or more sterigma, which in turn will form the conidiospores. Okay. Now I am coming to the sexual reproduction types. Again, gametophageal union occurs. It is the conjugation type. And you know, here most important thing here, I am just slightly showing you in the unicellular one. Then it is easier for you to understand also. Now then, similarly, plasmogamy followed by now Once plasmogamy occurs, it appears like this. Now this condition, what you call it? You can call it dikaryon. Having two nuclei of different type together, condition is called dikaryon. In ascomycetial and basidiomycetial fungi, the plasmogamy is not immediately followed by karyogamy. Plasmogamy is not immediately followed by karyogamy. So an intermediate stage is developed, and which is called dikaryophase. And this dikaryotic mycelium, eventually with these two nuclei, it gives rise to ascus structure, ascus. Actually, the whole structure will behave like ascus. Ascus may occur singly or a number of asci. A number of asci may aggregate to form ascocarp. I repeat it again. After plasmogamy, here in ascomycetial and you write it down, please. In ascomycetial and basidiomycetial fungi, plasmogamy is not immediately followed by karyogamy. As a result, a transitional phase is formed. As a result, a transitional phase is formed called dikaryophase. Possessing two nuclei, two nuclei from two different nuclei. Eventually from it, ascus is formed. Ascus, a number of asci may aggregate to form ascocarp. Already written here. Now in the ascus or ascocarp, first karyogamy occurs first, and it is followed by meiosis. So in these ascocarps, if it is an ascocarp, ultimately, first of all, what happens? Karyogamy occurs. And it is followed by meiosis. forming sometimes four and they are called ascospores. On germination, neomycelium. So I am repeating it again. During sexual reproduction, initially it is mainly conjugation type, and as you know here, at first union of cytoplasm of gamete occurs, which is called plasmogamy, and it is followed by karyogamy, but not immediately. After plasmogamy, a transitional phase is developed, having two nuclei of different types, and this phase is called dikaryophase. And the nuclei has two nuclei, when this nuclei will be termed as dikaryon, the cell. Dikaryophase stage then will give rise to ascas. Ascas may occur singly or sometimes a number of asci may aggregate to form a complex structure called ascocarp. Either on ascas or ascocarp, then first karyogamy occurs, and after karyogamy, then a zygospore is formed. Then it undergoes, within the ascas, it undergoes meiotic division, producing ascospores. And when the wall of the ascas is broken down, ascospores are liberated and will give rise to new fungal mycelium. This is about you need to remember two things here. First of all, in ascomycetial and basidiomycetial fungi, plasmogamy is not immediately followed by karyogamy, so that a transitional phase is developed. Secondly, you need to remember this transitional phase is called dikaryophase, which give rise to ascas. As ascas is developed, hence the group name is Ascomycetes. And you need to remember ascas may occur singly or they may aggregate to form ascocarp. Whatever it is formed, inside which first karyogamy is to occur. That means until ascas is formed, until or ascocarp is formed, karyogamy is delayed. So karyogamy takes place in the ascas, so that a diploid cell formed, which in turn undergoes meiotic division to produce the ascospores, which then germinate to form neomycelium. This is about the sexual reproduction type that is found in Ascomycetes. What to remember? I already told you. Now I think it is clear to you. Do you have any problem? Anyone, please? Prachi? Prachi, do you hear me? Important. Important Ascomycetial fungi. The names are, although you know, till now we are considering yeast under Ascomycetes, under fungi. But according to 5 kingdom classification, yeast as it is unicellular, it should not be under fungi, rather it should be under kingdom Protista. But still we are studying them under this. Common name is yeast. Saccharomyces cerevisiae. It is useful in many purposes. Baking industry, to produce vitaminized food, silk industry, baking industry, In silk industries also it is used. And lastly, brewing industry. Actually, which qualities of yeast is actually here utilized? Why yeast is used in different industries? Which quality is actually utilized? This is fermentation quality. It gets fermented easily, and which is used in different types of industries. Next comes to the next one. Next one is this is the most important fungus of this group, from which penicillin drug is obtained. And by extracting penicillin drug from Penicillium notatum, you know that Alexander Fleming discovered penicillin. And so this is most important now that penicillin is actually a kind of antibiotic drug. Next most important one is Claviceps purpurea. And from this LSD means, you remember, lysergic acid diethylamide. LSD is obtained from Claviceps purpurea, and so you need to remember this thing. It's a hallucinogen. Remember this thing. A drug developing falsehood is the hallucinogen. So it is the most important question coming in the entrance. Remember this thing. Claviceps purpurea is a ascomycetial fungus from which LSD is obtained. Remember this thing. Next one, Neurospora. Most important one. Neurospora is actually most important one used as in different experiments, used as experimental material in different experiments. So this is what about the Neurospora. And you know, Drosophila is used as experimental material in different
Sir,
experiments.
Drosophila is another fungi.
No. This is the most important thing. Drosophila means housefly. Neospora is used. Drosophila is generally Drosophila is very common. You will get it in class 12. T.H. Morgan performed his experiment on linkage by taking Drosophila as an experimental material because Drosophila can be reared easily in the lab. They can easily be grown in the medium. They can produce large number of progenies. They are actually male and female can easily be identified. A lot of characteristics are there. That's why it is easier for the scientists to rear Drosophila. Similarly, Neurospora fungus is behaving like Drosophila and act as experimental material in different genetic experiments. Biddle and Totham. Biddle and Totham, these two scientists proposed one gene, one enzyme hypothesis. And they performed their experiment by taking Neurospora fungus. So that's why Neurospora is also called, due to this similarity, you need to remember this thing. Neurospora is called Drosophila of fungal kingdom. Earlier fungus was under plant, then it was called Drosophila of plant kingdom. Earlier so you need to, now you see it clear now? Next important one is dangerous one, Aspergillus. It causes a disease Aspergillus is responsible for causing aspergillosis, a disease generally a kind of pulmonary or lung disease. Another important thing you need to remember, Aspergillus is also called weed of the laboratory, called weed of the laboratory. Why weed of the laboratory? Because this is the fungus found to contaminate most of the experiments. Different types of instruments are used, different types of living stains are used in the lab, and where it is found to contaminate. It is found to contaminate most of the laboratory material and utensils. That's why it is considered as weed of the laboratory. Remember this thing. It is called weed of the laboratory. Now, they are the most common examples. Another is one, Morels and Tafels. Most important one, morels and truffles.
Sir, it is
Morels
another
and
kind of
truffles.
fungus.
Huh? Not
Is
visible?
it, sir, is it another kind of fungus?
Yes, yes. Morels one type? Okay. Just a minute. Rub it out.
Sir, you may clear the board.
Hmm. Okay, I am repeating this. Yeast I discussed. Then penicillium I discussed. Then aspergillus I discussed. Then claviceps I discussed. Neurospora I discussed. Then now morels, truffles. So morels and truffles are actually edible. Similar to that of, I am showing you one thing, just a minute. Morels and truffles are actually edible fungi. Similar to that of mushroom. You see. Just a minute. Morels. Similar to that of mushroom. And this structure shown here is nothing but the ascocarp. Where a number of asci aggregate to form this. So in morels you can write ascocarp is edible, and this is the ascocarp. This is one morel, this is the other morel. Clear? Is it visible to everybody? Next
Yes, sir.
truffles. Okay. Next the truffles, I am showing you the truffles. Fruiting body. It is also a fruiting body, which is called the truffles. It is found inside. It is to take out from inside the soil. Okay, so these are the truffles. Is it visible to you? These are also ascocarp, actually they are to be taken out. Ascocarps are edible. Now we're proceeding towards the basidiomycetial fungi. Basidiomycetes. Point one. Mycelium is septate. Second one, modal structures are not found in the life cycle. Cell wall is made of chitin. Two types of mycelia are found. Most important. Primary mycelium, secondary mycelium. Primary mycelium means normal when the life cycle begins, the mycelium found, and secondary mycelium occurs again during sexual reproduction when plasmogamy is not immediately followed by karyogamy. A dikaryophase is developed here also, and it will give rise to, during that some growth occurs, and so mycelial structures are developed, and such mycelium developed in dikaryophase is called secondary mycelium. Is it clear to you?
Sir, please explain the secondary mycelium again.
Look one thing. During dikaryophase, as you I think already described about the dikaryophase, and in dikaryophase it grows to form a mycelium-like structure. Dikaryophase grows to form a mycelium-like structure called secondary mycelium. Are you getting me now?
Yes, sir.
And then it will form the basidio. Now, I am going to stop here, but I am not going to extend the time now today, but next day I will be giving up to 20 minutes extra time. Do you have any problem?